Hello, my name is Roger Sunahara. I'm a professor of pharmacology at the University of California, San Diego School of Medicine. I'd like to thank Biovia and Dassault Systems for inviting me to present some of our work on leveraging structural and biophysical data for studying mechanistic and mechanisms in disease and cellular signaling. My lab focuses on one particular type of signaling system, the G protein coupled receptor signaling. So G protein coupled receptors are cell surface receptors, and they function to recognize stimuli from extracellular sources and transduce those messages to the inside of the cell, where they can activate effectors such as adenylyl cyclase or phospholipase C, or even ion channels. And they activate these effectors through coupling to a small set of guanine nucleotide binding proteins, or G proteins. And these G proteins represent sort of like a switchboard, molecular switchboard. So activation of the G proteins and the receptors and the effectors leads to production of second messengers and also regulation of transcription, and results in a dramatic change in cellular responses. Could be contraction, could be cell migration, secretion, et cetera. So G protein coupled receptors are incredibly important for physiology. They're involved and critical for cardiovascular and cardiopulmonary function. They're extremely important in the brain, and many drugs that are used to treat neuropsychiatric diseases are target GPCRs. They're extremely important in immunology. Chemokines are the major, chemokine receptors are the major target for chemokines. And they're also responsible for 3 of your 5 senses. They're important for vision, smell, and taste. And without these GPCRs, you would be blind, you'd be anosmic, or you couldn't taste anything. And then more recently, GPCRs have been implicated in hearing, where the establishment and formation of hair cells and maintenance of hair cells are dependent on one particular GPCR. So what's interesting about the GPCR family is it's extremely large. GPCRs represent the 3rd largest family of genes in the human genome, and the largest family of membrane proteins in the human genome. And they're also very good targets for therapeutics. In fact, most of the nodes that I pointed to are in fact targeted by clinically approved drugs. There's a very nice review that was published about 20 years ago by Overington et al., where they had suggested that of all the drugs that are currently on the market, 27% of them target GPCRs. And then through the years, that has increased to the point where this year, at least 36% of all drugs currently FDA approved and on the market target GPCRs. And some examples of really more popular, maybe in the media, are some of these GPCRs, like the GLP-1 receptor, which is the target for GLP-1 and also the drug Ozempic. And you can see in this little bar graph on the side that the use of Ozempic and the number of prescriptions have just skyrocketed, as well as the profits from the pharmaceutical companies. Also noted in the media are opiate receptors. These are the targets for things like morphine, hydrocodone, and most devastatingly, fentanyl. And you can see in this picture that the dramatic deaths that have arisen from opiate overuse, any kind of opiate overuse, have skyrocketed through the years. GPCRs are so important for people, even things like antihistamines, like Benadryl and cetirizine, Zyrtec, are target GPCRs. They target histamine receptors. And then my favorite receptor, the beta-adrenergic receptor, the beta-2 adrenergic receptor, is incredibly important for treating things like asthma. Many people take albuterol as an inhaler to deal with their acute asthmatic attacks. And also the beta-1 receptor is a target for metoprolol or propranolol for use in cardiovascular ailments such as congestive heart failure. So I said the receptors that I like to study is the beta-2 adrenergic receptor. It's considered a prototypic receptor because it was one of the first receptors ever purified, ever cloned. And since it's a good target for therapeutics, there are lots of available drugs. So we know a lot about the beta-adrenergic receptor, and we can use that information to help us evolve our hypotheses on how receptor works. And as I said, the beta-adrenergic receptor, like many of the GPCRs, is incredibly important in physiology. So to activate a receptor, when a hormone binds, it activates the G protein. And when I say activation of the G protein, what I'm referring to is exchanging nucleotide. What the receptor does is it tickles the G protein and causes it to lose GDP. And inside any given cell, the cellular concentrations of GTP is really high, and that ends up binding to the G protein. And GTP binding to a G protein activates it, so they can now activate an effector like adenylyl cyclase. And so I've been studying this pathway for many, many years now and trying to elucidate how hormones activate G proteins. But for a long time, we were struggling basically with resolution as high as these cartoons. But it was really, we needed an atomic model. We needed something that could help us with drug discovery at a molecular basis to help us to understand how this receptor works, not only for drug discovery, but also for mechanistic studies. And finally, in 2011, we solved the structure of the beta-adrenergic receptor bound to a G protein. And this is through a long-standing collaboration with a very close friend and collaborator, Dr. Brian Kobilka at Stanford University. And here's just a rotating picture of the complex. And it's been incredibly informative to the whole field, the whole GPCR field, because it's provided the first snapshot into how a receptor could activate a G proteins. And its importance to the field was then recognized in 2012 by the Nobel Committee, and who awarded Brian and Bob Lefkowitz with the 2012 Nobel Prize in Chemistry. And the work cited in the Nobel Prize was actually this series of papers that Brian and I published in 2011, including the structure of this complex. And because of that, he actually invited me to attend Nobel Week, which was a lot of fun. So since this time, now hundreds of GPCR G protein complexes have been solved, and we've gained a lot of information in the field about how these receptors work. And we have now structures of many GPCRs in the inactive state, in other words, without a G protein, and now bound to a G protein. And this has had a huge impact on drug discovery, because now we know what is necessary for a small molecule to bind to a receptor to activate it, to make it a safer and a more efficacious ligand. The other thing that the structure revealed was pretty dramatic conformational changes in both the receptor, here highlighted in magenta, and the G protein sort of highlighted here in orange, but colored green and cyan. So here the alpha subunit of the stimulatory G protein, G alpha S, is the main partner that's engaging the receptor. And then when an agonist binds, it causes a dramatic conformational change in the G protein that results in separation of the alpha helical domain from the RAS homology domain. So the alpha helical domain in cyan and the RAS homology domain in green. And I'll show you an animation which really recognizes this dramatic conformational change which occurs when an agonist binds to the receptor. So here the alpha helical domain swings way, way, way far apart when the G protein is activated. And you can see the receptor itself, upon recognizing the hormone, undergoes this dramatic conformational change. So in my lab, we are really fascinated by this conformational change in the G protein, this really large conformational change. But it's hard to study. So we ended up collaborating with a very good friend of mine, Ron Dror, when he was a scientist at the David E. Shaw Research Institute in New York, and now he's at Stanford. But Ron was a specialist in molecular dynamics simulation. So Ron used computational methods, physical methods based on the physics of protein-protein interactions to study domain opening. And I'm going to show you one of the simulations in which Ron was able to look at separation at the alpha helical domain from the RAS domain as a function of time. And he can show that the G protein will spontaneously open. And coupled with our biochemical data in this paper we published in 2015 in Science, we're able to demonstrate that domain opening is absolutely required for nucleotide to fall out of the G protein or GDP release. But that opening itself doesn't trigger it. You need it to be accelerated by a GPCR. So the G protein, G alpha S, is encoded by a gene called GNAS. However, GNAS itself encodes two splice forms, GS alpha short and GS alpha long, and they have major differences in signaling. If you were to look at the expression of GS alpha short and GS alpha long in any given tissue, you can see that both GS alpha short and GS alpha long are both expressed in, for example, rat brain. So they're both expressed, but there's a little bit less GS long. And in collaboration with Emily Wheeler, who was a graduate student in Jean Yo's lab, and Shaylee Vora, who was a graduate student in Irini Papapatru, his lab, we did a really interesting study. So Jean's lab's interested in myelodysplastic syndrome. Jean and Irini are interested in myelodysplastic syndrome, which is a devastating disease in which there's a defect in differentiation of hematopoietic stem cells to different lineages, such as red blood cells, white blood cells, and platelets. And you can imagine if you have a defect, how devastating that can be for patients. And there are lots of genetic mutations which can predispose you to MDS and AML. And two of the most common mutations in two splicing factors, proteins that are involved in handling messenger RNA. If you have those mutations, you end up getting a dramatic overexpression of GS long and not short. So the two splice forms, now you get too much long. And this leads to a defect in ERK signaling, which I'll discuss in a second. But more importantly, it caused defects in hematopoietic cell differentiation. And so what is intriguing about this is that overexpression of GS alpha long, but not short, causes all these hematopoietic cell differentiation defects. So the ERK pathway, which is the extracellular regulated kinase pathway, is incredibly important for regulating cell growth and cell differentiation. And mutations in proteins that regulate ERK signaling have been implicated in disease. For example, RAS, and many people know about RAS mutations, which are found in cancer, is the primary upstream activator and regulator of ERK pathway. In fact, 20 to 25% of all tumors, human tumors, have RAS mutations. And in some tissues, such as pancreatic cancer, greater than 90% of those tumors have RAS mutations, and therefore effects on ERK signaling. And so through another collaboration with Jin Zhang here at UCSD, and again, with another very talented postdoc, Yonggung Quan, we were interested in beta adrenergic receptor stimulation of ERK, which has been known for many years to occur, but how it occurs was not known. And what we were able to demonstrate in this paper that was published in Nature in 2022 was that the beta adrenergic receptor specifically couples to GS long to regulate ERK and enhances ERK activation. not GS short, only GS long. So there's something unique about GS long versus GS short, some property of GS long that makes it not only um, um, have an odd activity, but it's involved in disease and ERK activation. So to summarize, mutations in MDS splicing factors increases GS long mutations expression, but doesn't affect GS short, and that leads to defect in hematopoietic cell differentiation. And this overexpression also results in dramatic effects on ERK signaling, which is a known pathway involved in growth and differentiation. And also that receptor catalyzed activation of G protein leads to ERK activation, but only through GS long and not GS short. Again, so there's something odd about GS long. So through a very interesting review that was published by Silvio Goodkin's lab, also my colleague at UCSD, where they actually sequenced several tumors, and they found that a surprisingly high number of tumors have mutations in G proteins, and specifically in GS, where about 4.5% of all tumors carry somatic mutations in GNAS. And of those 4.4, almost 90% of them are a single residue, arginine 201, to other mutations. And if you look in other tissues, biliary tract, testis, pituitary, that 4.4% goes up to almost 30% of tumors carrying these mutations. So it made us wonder, what is the effect of these mutations on GS long and GS short? And surprisingly, we observed that mutations have a profound effect on GS long and not on short. So on the right top panel is just a simple bar graph showing you that GS mutations in GS long in blue have a dramatic effect on the exchange rate or activation compared to GS short in white. Again, there's something odd about GS long. And then more recently, through postdoc in my lab, Daniel Meyer, incredibly talented and creative scientist, created an assay in which you can measure the domain opening. Remember the domain opening we saw in the structure? He can measure domain opening by measuring the rate at which an antibody binds to one part of the G protein, the alpha helical domain. And what he was able to show with this antibody was that cancer mutations are profoundly in the open state rather than in the closed state. And he also generated a novel biosensor, a GFP biosensor, which can sense the open and closed state. And he was able to demonstrate that cancer mutations like R201C stabilize the G protein in an open conformation. So again, there's something about cancer mutations, and particularly about GS long, that stabilizes the open conformation. So the structure and the biochemical evidence suggested that this open conformation is important both in activation, but also in disease. How about if we reapproach it? Is there a computational method which we can use to sort of mimic or predict the biochemical and biophysical data that we already collected? And so through using a collaboration we have with Andy McCammon, an eminent computational biologist at UCSD, and his postdoc, Haixin Wei, my graduate student, Cesar Tawfiq, a PhD candidate, tackled this question. Could you use computational methods to study the opening and closing dynamics of the splice forms in disease? And yes, I'll say you can. So the McCammon lab developed a really fascinating molecular dynamics simulations or computational approach, which is called kinetically accelerated molecular dynamics. And I'm not going to go into the details, but I'll just show you the data. So here are fluctuations of GS short wild type. And you can see how you can see it, some of the simulations, the alpha helical domains opening a little bit. If you put a cancer mutation in, it opens a little more, but not that much. Wild type GS long opens up more. But if you put the cancer mutation in GS long, there's a dramatic difference. So these simulations basically recapitulated all the biophysical and biochemical data that we collected on the unique features of the arginine mutations, the cancer mutations on GS long. And this is just another view of it. I'm sorry for all these graphs, but this highlights the fluctuations. In other words, how much the G protein is moving and parts of the G protein moves in short, long, wild type mutant. And in red is the mutant in GS long. And you can see how much it moves more compared to everything else. And it's the alpha helical domain primarily that's moving. Another interpretation is to measure the distance between residues that are either in the helical domain or the RAS domain in the closed or open. On the left is just a picture of the two residues I'm using and measuring the distance between in the closed on top and in the open on the bottom. And in the graph on the right, you can see that GS long carrying the R201 mutation rapidly opens. That's the tracing in white compared to the other G proteins. So we picked a classical signal transduction pathway, activation of the beta-adrenergic receptor, activation of the G proteins, regulation of adenylyl cyclase, and ERK, knowing that this is an incredibly important physiological target. And we used a lot of biochemical data and biophysical approaches to understand the behavior of GS short and GS long and the cancer mutations. But we were able to use computational methods to basically predict the same behavior that we observed in biochemical and biophysical data. We're also using other methods of molecular dynamics simulations for different projects. I'm not going to really talk about that, but it has been useful in unraveling detailed mechanisms about how G proteins are activated and how nucleotides actually falls out of the pocket. But I'll leave you with some food for thought. So it sort of begs the question of whether computational methods are advanced to the point, for example, using KAMD or molecular dynamics simulations, that we can create a virtual signal transduction pathway, one that can help us understand any disease-related effects. And you could dream, I guess, that you could use computational methods, particularly if you can incorporate some artificial intelligence, to to maybe virtually treat a tumor in culture. And, or even better, can you create a virtual patient with a known disease-related mutation and try to figure out whether there's a possibility of treating with a drug to treat this mutation, but computationally, can you model it? And even further dream, is it possible that maybe you can create a virtual twin of a given patient with for example, with a genetic defect? Is there something you can figure out on the computer based on a virtual twin of the patient that can help understand the mechanism of disease, but also the treatment of that patient? This work couldn't be done without fantastic collaborations as well as people in my lab. My lab, I talked about a lot of the work from Daniel Meyer, from Cesar, Nico, Villanueva, as well as technicians Mary and Geneva. And we've had fantastic collaboration with the Yo and Papa Petru labs, the Jin Zong at UCSD, and also Andy McCammon, who's really helping us with the computational methods. And I have a lot of friends and collaborators on the right side who have helped me throughout my career and on this project just by telephone conversations. And I also like to thank our funding mechanism, not only from the Department of Pharmacology, from National Institutes of Health, General Medicine, but also from Biovia, which has been incredibly generous and supported Cesar for all his work in using computational analysis. So I'll leave it at that, and I'll thank you very much for hearing my work, and that's it. Thank you.
Thank you, Roger, for advancing our understanding of cell signaling and molecular biology. Your groundbreaking research brings us closer to realizing the virtual cell, and that is absolutely foundational in the work that we're doing in creating the human virtual twin, linking biological mechanisms to predictive models of life and ultimately the human. Thank you.